anything to engage in public sort of yourself-biology, I decided to then and now that we need to have better and better tools for tracking the dissemination of the information, the chemicals, and the instruments. Here's just a snapshot from the trenches in the laboratory. We're trying to address what it is that people want when they say that they want organisms, genetically engineered organisms that do not spread in the wild. This is one of the many critiques of genetically modified organisms. And this is one attempt that we recently published where we make them genetically isolated, metabolically isolated, even if they, whether or not they're physically isolated from the environment, they cannot exchange genetic material successfully. So, some of the prediction. These were forward-looking statements where New York Times, nothing against New York Times, maybe, but back in 1903, they made the modest prediction that it would be 1 to 10 million years, a nice broad range for a flying machine. And even Wilbur Wright, who was much closer to the events than the New York Times, perhaps, was off by about 48 years in his estimate. And so we make such mistakes, and even within my field, my fields of computing and biology, we have these two trend lines, sometimes called Moore's Law. It's not really a law, it's a trend. It could deviate from the line at any point. The blue line, which is various biological things, the sequencing and synthesis, reading and writing DNA. It followed the electronics Moore's Law line at about 1.5 volts per year for many years, and so it seemed like, wow, this must really be a law. This covers general biology and electronics. But then we blew it, and we went way off the reservation, and it took about 6 decades to bring us down to a orbital genome, or more like 6 years. So we're down around $1,000 cost. We don't yet have $1,000 that you can go out and individually get genomes, but it's at bulk rate, that's where we are right now, which I think is cost effective today for a variety of things. Kevin Kelly documented how long it takes for new technologies in general, this includes crossroads and so forth, to be banned, and then the ban to be released. It's almost inevitable that technologies like really dangerous technologies like railroads and automobiles would be controversial. And then, but the time that it's taking before they become uncontroversial is shrinking. This isn't to say that we shouldn't have controversy and discussion. It's just where are we going? It's to describe where we're going. Now, these are three types of technologies I was intimately involved in. Each one of them had some sort of controversy and some sort of response. So the controversy being the bold one and the response. And I'm making a very casual, not well-documented argument that in some cases these were not deterrents to progress. They were good conversations that, if anything, accelerated the field. So the recombinant DNA was from 1974. This got a lot of attention, covers of Time magazine and things like that. And almost instantly, Genentech, Biogen, and Amgen, and a variety of other biotechs got investors. The investors said, oh, it's controversial, there must be something here, let's take a closer look. Similar thing happened in 1991 where the NIH actually started the controversy by patenting a lot of cDNAs that Greg Venter's group had done back when he had a small NIH lab. And the lawyers just said, this is what we do. We patent things that come out of the NIH labs. And then people said, we can't patent cDNAs. That's going to add much value to it. They're natural piece of DNA. We now see the cDNAs are patentable, but I would argue that there wasn't much value added to these. And the controversy resulted in a great deal of attention, and almost immediately that research moved out of NIH to Human Genome Sciences and therapeutics companies that I was helping out getting early sequencing going. Human embryonic stem cell, there were lots of restrictions in the United States especially. This resulted, I think, I can't say for sure, but I doubt that California would have cost quite that much money if there had not been that much controversy and frustration among the companies. one base pair out of 6 billion in your genome that you want to change. And in this case, this is kind of the earliest of these is taking it through clinical trials. Sangamo is for phase 1, phase 2 clinical trials on, and you delete or damage both copies of the CCR5 gene, and you end up with in the T cells, so of course you can take them out of the person, manipulate them, put them back in. That means there'll be no graft rejection, no need for immunosuppressants. And they now the ones that have taken up this zinc finger nuclease, this specific targeted nuclease, those T cells have been doubly knocked out of the CCR5 gene, are now resistant to the AIDS virus. And this can be done in patients that are resistant to AIDS, that already have full-blown AIDS. And so again, this is very promising for very severe diseases. We have a project to study personalized medicine in its broadest sense, ranging from carrier status, newborn screening, all aspects of modern human genetics that works quite well, to new personalized medicines, including microbiomics and complicated multi-factors, to get this multi-factor data set, this large data set about individual people, not about cohorts, but really lots of N-of-1 studies. We have this open access data that requires consent that shows people really what they're getting into in terms of the possibility, the probability that their data will be publicly available. We have stem cells for many of these individuals and a variety of tests that can be done. And the sort of stem cells that come out of this are a little piece of the brain initiative that you heard just a moment ago, where you can take these stem cells, reprogram them into neurons, and then they can have kind of neurophysiology, and it can be done in very short time frames. So we're getting to the point, another clinical issue, where we can not only manipulate human genomes, but we can manipulate human brains. We can either program them in situ with these innovative neurotechnologies, or we can build brains outside of the body and get this kind of spiking behavior on neural nets. This brain initiative was partially instigated by this group of 6 people that I was part of in 2012, and it's really remarkable how quickly it went from that semi-obscure forward-looking piece of neurons to an announcement by Barack Obama that we had a brain initiative. I mentioned nucleases that you can target. You can program to find a needle in a haystack of the human genome or other genomes. There are a variety of ways of making these nucleases very specifically. But they can be targeted not just in the human genome, but into wild genomes. We know that we're releasing genetically modified organisms into the wild for crops, for plants. There are a few animals that are being released into the wild. For example, Oxitec is releasing Aedes mosquitoes, which have been engineered to be essentially like nails that are sterilized by radiation, but in this case it is sterilized by genetics. That seems like a fairly safe thing, since the nails don't suck blood and they are sterile. It's a special case. But you can see that the communities are warming up the idea of releasing plants and animals into the environment. This would be the most extreme form of that, and as a consequence, Kevin Esfeld and Andy Schnitzler from the Katarussia and I published 3 papers very recently laying out this new form of gene drive. The basic idea goes back to Austin Burke in 2003 at least, but this is a CRISPR-based gene drive. It's so much easier probably to do, and might be something worthy of a cautionary tale, something worthy of being very thoughtful about, because it is so enticing and because it is so easy to do that even individuals might be able to do it. So now is the time to talk about it before we go much further. And since we published these papers, we've already now done experiments on gene drives. It's not published in yeast. We've done it in yeast because they have such a rapid generation time. This method is directed at sexual transmission, where you can correct spread. A desired package to allow population, or in this case, a laboratory population, exponentially, so all the offspring have it. And the yeast from the yeast has been it's been close to 100%. This is kind of a simple version of how it spreads. This is decidedly not like the greater mental piece that we saw a few slides ago, included in the pulse. This is something where essentially you introduce one or a few mosquitoes into the population that have the gene drive, and it spreads exponentially to all their offspring, subsequent matings, and so it is throughout the population. And it can spread packages such as resistance to the mosquito for malaria. So you're not actually hurting the mosquito, but you're making them so they no longer can be vectors for malaria. Or you could make it so that there's a little that if you only get transmission of the Y chromosome, in this case, you'll eventually result in decrease in the mosquito population. This is a little more technical of how what's happening behind the scenes, but you basically have the blue cassette in the upper left that is on one chromosome in one specific position. This is selfish DNA. It's not DNA that is wildly replicating all over the chromosome. Every chromosome is producing a huge burden on the mosquito, or whatever species you're dealing with, whether it's invasive or vector species. But it's in one place, in one chromosome, that you decided is the best place. And then when it's in the embryo, it finds that same place in the chromosome where the other parent doesn't have the cassette, and it will move the cassette over into that by making a cut, takes a double strand break, and the cassette moves into it. And now you either have both copies, all the copies of that particular chromosome looking the same, or perhaps you mutated. Either way, all the offspring that survive have the cassette that you want. And, forget the details of this, but the important thing is there are very sophisticated things you can do with this new technology, this CRISPR technology, that allows you to be very specific, target subspecies, So you have not only the species mating behavior as geographical barriers, but you have the genomic barrier that you can engineer so that you can make sure that the cutting is efficient and specific and even subspecies specific. You can do immunization either against gene drives or against natural components like viruses, and you can make reversal drives. I will make the argument that we are poorly adapted to our current environment. We did not evolve to sit all day and be exposed to giant amounts of really tasty food. And there are various opportunities that we have, which I would, certainly they're not eugenics, they're not even augmentation, and that we're dealing with diseases of civilization. They're augmentation in certain sense that we're putting things into our medicines or gene therapies that deal with things we wouldn't have to deal with in the past. But we respond to these in an emergency attitude because they are life-threatening. This changes again when we go into new environments, and our civilization keeps pushing itself into new places, such as space. In space, we have problems with osteoporosis due to lack of gravity. We have extreme radiation problems. Now, some of these have corresponding problems on Earth. There are problems with inner ear. There's problems with special stressors that occur and so forth. When we deal with these situations, we worry about what percentage space. So how can we possibly understand something as complicated as height? When there are now 700 genes infected, they have very tiny effect sizes, which is important. And you find it from projects like the Personal Genome Project, where you can correlate traits with genomes. It's looking at the extremes, where you look way out at the shortest and the tallest people, as an example of many different traits, both medical and non-medical. And you see out of those extremes, you get things with metastatic genes and variants that are so powerful that they dominate metastatically all of those small effects, including some small environmental effects and small genetic effects. And these suggest cures as well, or treatments that will influence these very complex genetic and environmental traits. So if you give growth hormone to a young person, maybe even at an age where growth should be stopping, growth will continue and their stature will approach whatever stature goal they've had. So this is an example of something where you have what looks like a very complex genetics with fairly simple consequences. Anything about medicine is simple. You also have this. You will often have cases where you have one person on the planet at any given moment where you run it. You don't have to be statistics here. You have a cohort size of one. Nevertheless, you can prove human in vitro organ models, and these you can consider rare protective alleles. These are things like in this case myostatin. You get a double null, meaning missing both copies. And at more or less at birth, the child had high musculature, as you can see reflected in the animal models. And there are many of these. All of these are, none of these are unmitigated protective variants that have no downside, but they have interesting possibilities. Some of these I mentioned in the context of imagining that they could be useful to deal with diseases of civilization or new environments in space. So, for example, there are LFP variants. This is a heterozygote that extra strong bones or missing growth hormone. You might have smaller people in space. You have interest in pain. You have odor might be involved in space. You can have multivirus resistance, low coronary artery disease, and so forth. These are floating around in our population. Many of them minus or minus are double nulls. These are genes that are conserved among all animals, but they're missing in human beings, and they're not only alive, in many cases they're thriving. A possibly extreme example of this are people who live past 110 years. This slide is not an advocacy of smoking and drinking to excess, but the point is it's probably not solely that they have a great environment. They may actually have some exceptional genes, and so we're studying their genomes and trying to test hypotheses about them. Wrapping up here, we actually study biospheres, how our environment interacts with our genetics in more detail. I've already mentioned toxic radiation. There are organisms that are very different related to us, which are radiation resistant. And you might say, how irrelevant can that possibly be? How many mutations would that take? Well, it turns out you can change with as few as 4 mutations an organism to be 100,000 times more radiation resistant. Whether this applies to humans is still unknown. And I'm just going to end on this very playful and speculative slide. This is a typical operating room. We will have to make a decision as we go into new environments outside of Earth, whether we want to drag along with us all our competence. We can or we can't. This is, I consider that part of genome engineering is how we interact with the huge part of our genome, which is in our microbiome. And I mentioned that there are people that have sensitivity to pain. You can engineer this such that it can be turned on and off. And so rather than going into general anesthesia with this risk or opiate type analgesics, you can have something like this. So I thank you and time for questions, I hope. So they asked me to talk about my life and sort of how I got to be a scientist. So I'll take you through some stuff here in a moment, my life plan. So my family life, I was the youngest of 4 kids. I had 3 sisters. We were not a wealthy family. We were actually pretty poor, strapped for money. We didn't have much money. You don't need to come from a good family. You don't need to have money. You just need to be determined to make something out of your life. The first thing that I was interested in were dinosaurs. I didn't know they existed. I went to a small Catholic school. They were teaching a lot of evolution back then. And so I was just amazed that there had been these giant animals that lived millions of years ago, and that we could study them by looking at their bones and learning about them. So this is fascinating. I had no idea running around, playing with toys. He learns that there's this ancient history, so this life has gone on for millions before the Jesus showed up. And things are very small, and things are very large. And so it just started changing the way I viewed and perceived my world. And I really different, because it's just amazing that you can just, everything is made something smaller and smaller and smaller building blocks. Even the atoms are made out of different building blocks. So I got a chemistry set and did like a chemistry experiment that basically then I perfume, smell, soap, and turned things colors and more blew up my kitchen. I had a chemistry, and that sort of set me on the life of science. I decided to go to college. My teacher thought I should go to college. No one in my family had ever gone before, but I was able to get into the University of Illinois. If you try hard, and like I did, you can make the most out of what you have. So I graduated college. I went to grad school in biology at MIT, and even though I wanted to be a chemist, at my senior year in college, I learned about something called DNA, and then you could do recombinant DNA and switch it around and recode DNA. And I thought, wow, I want to do that. So that's how I got here. And now I want to tell you a little bit about biology and the way I think about it. I think that a lot of people think biology is way too complicated. It's much easier to comprehend with one simple realization, and that is that a cell, which we're all made of, with billions of cells, is much like a city. So a city is like a cell. And I'll give you some examples of how that is and how cities run and how they run like a cell. So for cities to run and be maintained, they need many different types of machines. Cells are also run and maintained by machines just like cities. The only difference is that their machines, being very small, are not made out of metal and plastic. They're made of proteins. Cells have motors, they have scissors and wheels and levers. They even have things like duct tape to stick things together. They have garbage disposal and incinerators. They have socket wrenches. They have delivery trucks. They have all the sorts of equipment and machines that you just look around and see every day. They said they're made out of proteins. And so duplicating cells, like duplicating a small city, in order to do that, you need blueprints. And in the case of a cell, the blueprints are the DNA. Now, the part of research that I got involved in is trying to understand what cells, how cells try to make sure that blueprints don't get changed. If you don't do this, you get mutations, and mutations can cause developmental disorders in children. They can cause cancer in adults and children. So you really want to get the blueprints right. You start changing the information in the cells. The cells start doing different things. So I discovered something that we call the DNA damage response, which is like a command and control center. And it flips a lot of switches on a lot of proteins and tells them to change, which is so important to duplicate your DNA right. So I hope that makes it a little bit simpler. I think that if you set your mind to something and you apply yourself, you can accomplish your goals no matter where you start from. They don't have to be science goals. They can be any kind of goal. But it's all about you. It's not about your family. It's not about your friends or what other people think about you. It's what you want to do with your life. And you can do whatever you want with it. So I'll stop here. Thank you. Historically, scientists have been able to also retrieve domesticated plants and animals, but have not been able to do this to wild populations. Here we use mosquitoes as an example to explain why most genome alterations designed by humans don't persist in nature, and how a recently proposed technology could change that. The transgenic mosquito, Santa Blue, has an altered gene inserted into one of its chromosomes. The plant mates with the wild-type mosquito. Each parent contributes one copy of each chromosome to their offspring. Thus, only 50% of offspring will carry the altered gene, while the other half will inherit the wild-type version from both parents. Even if the altered gene doesn't reduce the likelihood of these mosquitoes surviving and reproducing, it may persist at a low frequency in the ocean of wild mosquitoes, or it might go extinct after several generations, especially unlike inheritance. This process is what keeps us united around mosquitoes, prevent them from transmitting species such as malaria or dengue. A team led by Kevin Esler at the Research Institute, the Harvard Medical School, and Harvard School of Public Health has now outlined ways to gene drives that can improve the odds that almost any altered gene will be inherited, potentially allowing them to spread through the wild populations. The proposal relies on the CRISPR-Cas9 system, human genome editing technology co-developed by the researchers at Leeds. Gene drive mosquitoes carry both the altered gene, the genes for the Cas9 enzyme, and several guide RNAs that tell it where to cut. When passed to offspring, the guide RNAs direct Cas9 to cut the wild-type version of the gene inherited from the wild-type parent. Each cell then copies both the altered gene and the drive that it prepares to damage. Because the mosquito now has two identical copies, one on each chromosome, all of its offspring will inherit the alteration in the gene drive. The same process will be repeated in subsequent generations, causing the alteration in the gene drive to spread through the population. Recent development in nanotechnology allows the building of three-dimensional structures using DNA bricks. These bricks work a lot like interlocking Lego pieces. Each of these new nanobricks is made up of a single strand of DNA, and each strand is 32 nucleotides long. You can think of the strand as a Lego brick that has two pins and two holes. The pin of one brick is able to plug into the hole of another brick. held before we allowed them to start the shuttle. We've built over 100 different shapes in the lab from the 1,000 Marshall campus. Here's a computer-generated view of some of these shapes. And here are microscopic images of the actual shapes as seen from above.